Hello, my name is Alfredo Berzal-Herranz, and I would like to thank the organizers and the scientific committee for inviting me to present a keynote presentation in this first International Electronic Conference on Medicinal Chemistry. I am a researcher at the Institute of Parasitología y Biomedicina López Neira. This institute belongs to the Spanish National Research Council, CSIC, and it is located in the city of Granada, Spain. The subject of my talk is application of RNA Aptamers to the control of the function of the Hepatitis C Virus-CRE region. I will start my talk giving you a brief overview of the Hepatitis C virus and the Aptamers technology. Following, I will focus on a recent work of my research group exemplifying the usefulness and potential of the Aptamers as antiviral agents. This work has been co-directed by my colleague Cristina Romero-López. The Hepatitis C virus is an enveloped single-stranded RNA virus. It is the prototype of the Hepativirus genus belonging to the family Flaviviridae, and it is the etiological agent of the Hepatitis C. This infection affects around 3% of the world population. This means more than 180 million people. Therefore, it represents an important worldwide health problem. In about 80% of the patients, the infection becomes chronic and it causes a high incidence of cirrhosis and hepatocellular carcinoma. Currently, there is no available vaccine, and until very recently, the treatments have shown a very limited efficacy. This is mainly due to the high genome variability, which is a feature of the RNA viruses. This high variability is the main cause of the rapid appearance of resistant variants to drugs and the easy escape to the treatments and the immune system. This high variability is a consequence of the lack of proofreading activity in the viral RNA polymerase. But importantly, within this highly variable genome, highly conserved regions, both in sequence and in structure, have been identified. This great conservation means that these regions must play an essential function in the viral cycle. These regions that are defined as functional RNA domains have acquired a great importance for understanding the molecular mechanisms of the virus. This figure shows the secondary structure model of the 5' and 3' ends of the HCV RNA genome, showing the different functional RNA domains that are responsible of the essential viral functions like translation, replication, and encapsulation. At the 5' end, we can see the iris region, which is responsible of the initiation of the HCV translation. At the 3' end, we can distinguish the 3' X tail, which is essential for the viral replication. Another important functional domain is the CRE region, included at the 3' of the coding sequence. It consists of 3 stem loop motifs named 5BSL 3.1, 0.2, and 0.3. These domains exert their functioning by contacting with each other or by contacting with other domains and creating a complex network of long-distance RNA-RNA interactions. They also recruit both viral and cellular factors. I would like to fix your attention on this CRE domain. It stands for Fish Replication Element. It is the object of the work that I am going to present here. CRE is essential for replication, and it has been involved in different long-distance RNA-RNA interactions with RNA elements of the 3' X tail and with the alt domain. Few years ago, we saw that it is also involved in a long-distance interaction with the 3D element within the iris region, regulating its activity as initiator of the viral translation. Due to these essential functions, interfering with the functioning of RNA domains offers a potential means of developing anti-HCV strategies, being nowadays the Aptamers excellent candidates for targeting highly structured RNA domains and then for the development of antiviral agents. Aptamers are oligonucleotides that bind specifically and with high affinity to a ligand molecule. In 1990, two independent publications from the groups of Dr. Jack Szostak and Larry Gold defined the basis for the development of what we can call the Aptamer technology. The group of Jack Szostak described the isolation of RNA oligonucleotides that were able to efficiently bind to organic dyes and used the term Aptamer to name them for the first time, whereas the principles of the general strategy for the selection of aptamers, that is known as selects, were defined by the Larry Gold's group. The potential of this technology was fairly soon envisioned, and the potential clinical application of aptamers for a great variety of diseases have raised much interest, and it was in 2004 that the FDA approved the first aptamer to be used as a specific drug, in particular for the treatment of the wet age-related macular degeneration. It was commercialized as Macugen. It is a chemically modified and pegylated RNA aptamer. Throughout these 25 years since the first publications, a large number of reports have described the successful selection of aptamers against a large variety of target molecules. Molecules of different nature and different size, that goes from small molecules like ions, amino acids, nucleotides or organic compounds to macromolecules like proteins, nucleic acids, and even against virus and whole eukaryotic cells. And although different authors have introduced variations in the selection strategy imposed for the application they were developing, all of them respond to the original selects procedure defined by Larry Gold's group. This general strategy is called selects, where selects stand for systematic evolution of ligands by exponential enrichment, which consists of iterative cycles of synthesis, binding, positive selection, and amplification steps over a randomized oligonucleotide pool. Through the consecutive cycles, the resulting population is enriched in those molecules able to bind to the desired target molecule. Those are the aptamers. The selects procedure is represented in this figure, exemplified for the selection of RNA aptamers against to a molecule that is fixed to a streptavidin column. Usually the selection starts from a large population of RNA molecules, usually resulting from the randomization of 20 to 40 nucleotides, and they are obtained from the transcription of chemically synthesized DNA oligonucleotides. The variation in the system is introduced at the level of the synthesis of the DNA oligonucleotides, and this heterogeneity is in all moments determined and controlled by the researcher. The random nucleotide region is flanked by fixed sequences called primer binding sites that are used during the selection and amplification of active binders. We have applied this strategy to the selection of RNA aptamers against the HCV-CRE region. So we started with an RNA population of theoretically more than 10 to the power of 18 variants, resulting from the randomization of 30 nucleotides. And this pool of RNA was challenged for binding to a genomic RNA fragment containing the CRE region fixed to a solid support. After 6 rounds of selection, all selected molecules could be grouped in 5 groups defined by the existence of a selected sequence motif, which was complementary to a specific sequence within the target RNA CRE. These sequences are predicted to be involved in the initial binding of the 2 molecules. It is important to notice that the group 5 binding site maps just outside of the CRE region in a stem loop domain called 5BSL 3.4 that contains the translation stop codon. In this table, we have included a set of 44 different aptamers. The numbers indicate the group to which each sequence belongs, and interestingly, most of these sequences contain more than one consensus sequence. It means that they can be classified within more than one group. As a first screening of the selected aptamers, we tested their capacity to interfere with the HCV replication. For this purpose, cells derived from a hepatoma cell line containing a subgenomic replicon construct were transfected with the different aptamers. At 18 hours post-transfection, total RNA was extracted and the relative amount of viral RNA was quantified by RT-PCR. As it can be seen, approximately 35% of aptamers promoted a higher than 50% reduction in the replication levels, and 4 out of them, the aptamers P749, P696, P689, and P6-103, showed inhibitions higher than 75%, reaching up to 90-95% for the aptamers 96 and 103. These 4 aptamers were selected for further analysis. Since the activity of the aptamers is directly dependent on their three-dimensional folding, we wanted to know whether they share a common structural motif that could define a functional domain within them that might explain their high inhibitory activity. The prediction obtained with the TurboFold tool yielded a common secondary structure for all of them, in which the constant sequence corresponding to the ones used as a primer binding site during the selection process appeared as single-stranded tails flanking a unique stem loop domain that contains the selected consensus motifs, which are at least partially exposed in the apical loop of this structural element. This folding gives the idea that the functional unit is restricted to the stem loop, which would be responsible of the efficient interaction with the target site within the CRE region. Since it is well known that the CRE region recruits the viral RNA polymerase, known as NS5V protein, we wanted to explore whether the aptamer interference with the replication process might be due to competing the binding of the viral RNA polymerase. And as it can be seen in the figure, aptamers P6, 89, 96, and 103 efficiently interfered with the binding of the viral RNA polymerase to the CRE RNA in a concentration-dependent manner with EC50 values in the nanomolar range. Interestingly, the aptamer P749, which theoretically targets the adjacent stem loop that contains the stop codon, showed almost no competition activity. Finally, we tested the putative effect of these aptamers in the viral translation, since their target sites were included in motifs involved in the regulation of the HCV translation. In a similar experiment that the one described previously for assaying the effect in replication, but using a subgenomic replicon encoding the firefly luciferase under the control of the HCV iris, we observed a 50% enhancing of translation at 4 hours by the aptamers P6, 89 and P749, which is maintained in the time for the last one, whereas no effect was observed for the other 2 aptamers. This effect of the P749 aptamer might be explained by a direct interaction with sequences close to the stop codon, just at the 3' of the CRE region, as can be deduced by the sequence complementarity between both the aptamer and the target RNA. Our data confirm the potential of the CRE region as antiviral target and support its function in the viral translation and in the recruitment of the viral RNA polymerase. Our results also point to the domain 5VSL 3.4 as a novel efficient antiviral target and open a new field to investigate the role of this element in the consecution of the viral cycle. Our results also support the idea that the use of aptamers as molecular tools is a feasible strategy for developing antiviral agents and for investigating the functionality of RNA domains in viral genomes. And finally, I would like to thank all of you for your attention, and I'll be willing to answer any question at the indicated email address.